It's my pleasure to introduce Mark Chong today. I think Mark will be probably known to many of you, particularly because of his pioneering work on microRNAs. But just a brief background, Mark is a graduate of Melbourne University and did his PhD here at WEHI in the old Autoimmunity and Transplantation Division with Tom Kay, and then went to New York and worked with Dan Littman for several years. When he was here at WEHI in his PhD, he studied primarily the role of SOX1 in regulating T-cell differentiation and also in regulating responsiveness to cytokines in pancreatic beta cells in type 1 diabetes pathology. And then with Dan Littman in New York, he did a lot of things actually. He looked at the epigenetic control of CD4 expression. He studied transcriptional regulation in regulatory T cells. And then, but most of all, he really did a lot of work on microRNAs, primarily by knocking out the enzymes that are responsible for the biosynthesis of microRNAs, DROCHA and DICER. And in fact, he gave a seminar here. He's given 3 seminars here since he's been back. He gave one here just over 2 years ago or so on this subject, and he's giving another one today. So where are you? Whoever's running the program here, I think Mark actually exceeds the rate of seminar presentations. He's way ahead of average. In any case, it's great, Mark, to hear where this is going and all the collaborations that you've made. So,
thanks, Len. So I think there's probably quite a few of you here who've heard me speak before, and those of you who have probably used to hearing me talking about T cells and most frequently about microRNAs. But being a Wednesday seminar and all, I thought I'd do something a little bit different today, and really present our work at a more basic biological level, really approach it in a way that potentially has more implications in a broader biological sense, but of course still taking some examples from the immune cell types that we work on. So specifically I'm going to be talking about RNase 3 enzymes, and as Len mentioned, these, or at least 2 of these, are now synonymous with the microRNA pathway, and I'll come to these in a moment. But RNase 3 enzymes were actually discovered long before microRNAs were. The enzymes were first discovered in bacteria, and for example, shown here is the RNase 3 enzyme. It's called RNase 3 or ribonuclease 3. This is from Ecophex aeolicus, a small bacterium. It's quite interesting biology in this bacterium, but that relates to RNAi. But for the purposes of this talk, this enzyme is actually a small protein that works as a dimer. There's, shown here, there's one molecule here, one here. And what's important about this protein is it binds to double-stranded RNA. So sandwiched between the 2 monomers is this double helix of a double-stranded RNA. So bacterial RNase 3 is a very simple protein. It's short, contains 2 simple domains. The first one is a double-stranded RNA binding motif, so that binds double-stranded RNA. And it has an RNase 3 domain, so this is where the name of the family comes from. And RNase 3 domains cleave double-stranded RNA. So that's bacteria. Now if we move on to animals, we now bring up 2 much more familiar proteins, DROCHA and DICER. So DICER, these 2 proteins are much larger, you can see, many more domains. And these, until recently, were the 2 enzymes that we knew of in animals. In fungi, there is, depending on the species, there is 1 or 2 DICER genes. In plants, there are 4 DICER genes, but neither have a DROCHA gene. So this DROCHA enzyme is actually specific to animals. So until recently, really only until the last couple of years, these 2 enzymes were the only 2 RNase 3 enzymes that we knew of in animals. However, in a couple of years ago, we performed some homology searches to look at potential proteins with common sequences to these RNase 3 domains, and we pulled out a putative 3rd RNase 3 protein. We currently don't know what the function of this is, and Janet Yeo in the lab, a PhD student, is now working on trying to understand what this is doing. But interestingly, this protein is much more similar to the bacterial RNase 3 compared to these 2 proteins. Now, I'm not going to be talking any more about this, but hopefully you'll hear from Janet at some point in the future. So I'm going to focus on these two. So I'm going to split the talk up into three stories. Most of my time will be spent on these first two stories relating to Drosha. And then I'll see how I go with time, but in the last 10 minutes I'll touch on this last story that's related to DICER. So the three stories are some work we've done on the mapping of the structures of microRNA genes, a story about a function of Drosha that is nothing to do with microRNAs, and then this DICER story is also one that is it's a function that has nothing to do with microRNAs as well. So let's start with the first one. So this story was just published recently, and it's work that was done primarily by two former research assistants who worked with me. Francis Corrigan was responsible for most of the computational biology, and Ken did a lot of the bench work. So I'm going to tell you basically some stories of some of the findings we've made in our attempts to map the structures of microRNA genes. So microRNAs, I'm sure everyone here would have at least heard of microRNAs. They're small, 22 nucleotides on average. They function as regulatory inhibitors of messenger RNAs. They basically target the 3' untranslated region of messenger RNAs. And shown here in this example is Lin4, which is the first microRNA, which was the first microRNA discovered, and that was found in C. elegans, the nematode. And as you can see, it recognizes to multiple sites within the 3' UTR of its target, Lin14. One thing you'll notice that is there's not perfect matching. There's actually lots of mismatches throughout and always bulging. And this is a hallmark of microRNA targeting in animals. So the result of this targeting is translational repression, inhibition of translation of its target, and ultimately degradation of that target. So these tiny microRNAs are actually derived from longer precursors. They're derived from these stem loop structures embedded in longer RNAs. And the processing and maturation of that microRNA is dependent on our two enzymes of interest, Drosha and DICER. So the first step in microRNA maturation is the release of this stem loop structure, and this occurs in the nucleus. This stem loop structure then gets exported out into the cytoplasm, where it's processed by the other RNase 3 enzyme DICER, which cleaves off this loop, and it releases the mature microRNA from one or either arms. So we've been working a lot with microRNAs, as Len alluded to, for many years now, and we had been interested in the role microRNAs play in controlling the differentiation of particularly the T cell lineage, but also of other hematopoietic cell lineages. And one thing we noticed through our studies was just how dynamic the expression of microRNAs were. They were actually really quite dramatic, some of the expression changes that occurs. So going from a hematopoietic stem cell, an LSK cell, through to a mature T cell or B cell or dendritic cell, we find that the microRNA expression levels can change by as much as 3 orders of magnitude. So really to us, that suggested to us that not only was this pathway very important, the actual processing, there must be something very important about the regulation of the transcription of microRNA genes. And we were obviously interested in understanding that level of regulation. Now the problem was microRNAs, for most of the microRNAs, all we knew was the sequence of that mature microRNA. And from that sequence of the microRNA, we can pinpoint where in the genome the microRNA is derived from. For example, MIR210 is derived from this position. However, that's all we knew about it. We didn't know anything about the structure of the microRNA, unlike, say, neighboring protein genes, which we can see the exonic structure. So this is simply where that mature microRNA is from. Now, the reason for this was that we never were able to detect this primary transcript, the RNA that came off the gene itself. And the reason is this primary transcript is rapidly processed by the Drosha complex to enter the microRNA pathway. And without this RNA, primary RNA, we were never able to map the structure of the microRNA genes. So obviously, in order to be able to detect this primary microRNA transcript, what we needed to do was to block this processing. And to do this, we decided to knock out the Drosha gene in order to block this first processing step and allow the accumulation of this primary microRNA. And that's exactly what happens when we do that. So the data I'm going to show you now are mainly for CD4 T cells, but the way we've done this mapping could have applied to any other cell type. It could have been done to a liver cell, a B cell, or any other cell for that matter. And certainly others are beginning to do that. So firstly, this top panel is detection of the primary transcript using Qpcr. So you can see in the control, this is the level of the primary transcript. When you knock out DROSHA, there's a nice accumulation of the primary transcript, but not with DICER, the downstream enzyme. Same with this gene. Down the bottom is another way of looking at this accumulation of the primary transcript. This now is RNA-seq, so next-gen sequencing. This here is the location of the MIR29 cluster. So there are 2 microRNAs at the spot, 29A and 29B. You can see in the wild type, there's very little RNA at this locus, but when you knock out DROSHA, there's now a nice accumulation of the RNA in the locus. So that's great. We now have the primary transcript accumulating, but we still needed a bit more information in order to map the structure of the gene. So to do this, we employed mapping of 2 histone modifications. And these provide some marks of features of actively transcribed genes. So we look for the deposition of trimethylation on lysine 4 in histone 3, as well as trimethylation of lysine 36 on histone 3. So these are present on actively transcribed genes and allows us to map the structural features of that gene. So they deposit very differently. H3K4 is deposited specifically at the promoter, while H3K36 is deposited throughout the gene body. So by combining these 2 histone marks, we're able to delineate the boundaries of an actively transcribed gene, and when we combine it with the RNA, we can now start mapping the structures of these microRNA genes. So let's start with a couple of simple examples. These are 2 small microRNA genes. The top example is the MIR17 cluster, which contains 6 microRNAs across here, 17 to 92A. And this bottom is a simple microRNA gene containing a single microRNA, MIR142. So in the top lane of each is the H3K4, which should mark the promoter, 36 should mark the gene body, and here's the RNA. And in both cases, you can see, based on the size, the scale here, you can see these are basically small genes in the order of 1 to 2 kilobases. It's basically RNA transcribed throughout this locus, and what happens is this RNA simply enters the microRNA biogenesis pathway. It gets processed by DROSHA and so forth. So these are what we would term simple genes. But of course, microRNA genes can be a lot more complex, and everything in biology is never that simple. So, I failed to mention, about 1/3 of microRNAs are actually derived from the introns of other genes, so protein coding genes. So the regulation of those are intimately controlled together with the host gene. But the majority of microRNAs are these independent genes that we're interested in. So here's a couple of examples of large genes. The top one again is the MIR29A, 29B gene, and this turns out to be the largest gene that we've come across so far. So what you will notice is it's a lot more complicated, the structure. And if we take the entire genome interval from the most distal promoter all the way through the 3' end of the RNA, we find that this interval here encompasses something like 170 kilobases. That's 170 kilobases for 2 little microRNAs that are 22 nucleotides each. This one is another example. This is LET7BC gene. From the distal promoter all the way through to the 3' end, it is about 60 kilobases. And we see lots of these examples. Here are a few more at the bottom. You see some of them are over 100 kilobases. So the other couple of features we noticed was these multiple peaks in H3K4, and these indicate alternately employed promoters. So one starting here, and one here. So alternate promoters are, of course, a feature of protein coding genes as well, which allows for higher levels of regulation. And finally, the other interesting feature that we noticed was these gaps in the RNA sequence. These gaps suggested to us that there might be splicing going on. And again, splicing is a hallmark of most protein coding genes. So to confirm that we are really seeing splicing going on, we use primers flanking these putative introns and then perform PCRs amplified in sequence. And sure enough, we could confirm all the splice junctions. So what we have here are most of these microRNA genes being very large, many of them more than 100 kilobases. They can be transcribed by multiple promoters and to generate these long transcripts which can be extensively spliced. So certainly a lot more complex than we had ever imagined regarding microRNA genes. So why such long transcripts just to generate these tiny microRNAs? So we're basically discarding 99.9% of the RNA Could this be spurious transcription? I don't think so. I mean, it is possible, but I like to think that all that transcription is functional in some way. There must be some reason for generating all the RNA, even though most of it's discarded. One possibility we're thinking about is that the flanking RNA, the single-stranded RNA that's around the stem loops, may help regulate the microRNA processing in some way. And certainly there's good evidence, at least for the RNA immediately flanking the stem loops. It's possible that distal sites may contain cis elements that may be recruited that ultimately regulate the maturation of the microRNA. Again, we don't really know whether this is occurring, but it's certainly a possibility that we'd like to explore. And what about the splicing? Why are we seeing so much splicing going on? So in protein coding genes, you know that splicing is a mechanism by which you assemble the exons to generate a functional open reading frame in order to generate a protein. Or in the case of alternately spliced transcripts, it's to generate different protein products. But in the case of these microRNA genes, the spliced RNA is eventually discarded anyway, so it has no function. Again, we're not really sure what's going on. Potentially, splicing may help with the transcription of very large genes, and there's certainly some evidence that splicing can regulate the processivity of RNA polymerase II. Ultimately, we just don't know, and really we still have a lot of work to do into understanding what's going on with these two. And finally, just another layer of complexity. It turns out that microRNA genes can overlap with protein coding genes. And we picked up something like 6 or 7 examples so far. This is one example here, MIR21. So this MIR21 is actually located within the VMP1 gene, which is protein coding gene. And until now, it was thought to be derived from this VMP1 gene, and therefore its regulation coordinated with VMP1. So the VMP1 promoter is actually all the way over here somewhere. So when we look more carefully at the structure of this gene with MIR21, it turns out that there is another promoter here in the second last intron of VMP1. So we suspected that it was this promoter that was actually generating this microRNA. And when you look more carefully at the RNA level, you can see how different the RNA is in the Drosha knockout compared to the control. So what we have here is actually the microRNA transcript starting in the second last intron and going through at the same time. So in other words, we have 2 genes overlapping each other, but 2 different promoters driving transcription along the same area. And this confirms that this is all Drosha dependent. So these microRNAs now are becoming more common, and I suspect that many more of those microRNAs that were thought to be regulated together with the host gene may in fact be regulated separately. Okay, so that's pretty much all what I'm going to show you for that first part of the talk. What I showed you was that primary microRNA transcripts can really only be detected if you disrupt its ability to enter the microRNA pathway. In our case, we've deleted Drosha and therefore prevented that first step in microRNA processing. This allowed us to map the structures of microRNA genes, at least the ones in the cells that we looked at. And from this work, we found that microRNA genes, on average, encode 2 stem loops. But this can vary from anything from 1 microRNA up to several dozen microRNAs. Some microRNA genes are small and simple, simply generating a single transcript, short transcript that enters the microRNA pathway. However, the majority of microRNA genes appear to be large and complex structures containing multiple promoters and extensively spliced. And finally, I showed you that microRNA genes can overlap with other genes. All right, so that's the first story. The second story I will talk about is also about Drosha. And this is a finding, this project I started quite a long time ago while I was still a postdoc with Dan Littman at New York University. And this was a project where we were just trying to understand the function of these enzymes in controlling microRNA biogenesis. And it turned out that Drosha was doing more than just microRNA biogenesis, and that's what I'm going to tell you about. So this work is still ongoing, and we're still very much interested in this, and one of my students, Tim Johansson, is still working on this question here. So let me just remind you, microRNA biogenesis requires the 2 enzymes Drosha and DISA. So if microRNA biogenesis, if the microRNA biogenesis was the only things these 2 enzymes did, you might expect that knocking out Drosha and knocking out DISA would give you basically the same phenotypes, or very close to the same phenotypes, because ultimately both of them would prevent the generation of microRNAs down the bottom. and that's what would cause the phenotype. So in thinking about this, we decided to do a whole lot of comparisons between the knockouts of Drosha and Dicer, and of course we found things that we didn't really expect. But first off, an example of what we did expect to find. So this is an experiment where we knocked out Drosha and Dicer in CD4 T cells and looked at the outcome of that knockout. In this experiment, this is a SciLike experiment, which is a proteomic profiling experiment. We're simply just looking at the protein expression levels in these cells. I think in these experiments we measured something like 2,500 proteins, but this is just a way to show what the phenotype is. So on the x-axis is the change in protein expression in the Dicer knockout cells, CD4 T cells, and the y-axis is the change in protein expression in the CD4 T cells that have knocked out for Drosha. And when you do the side-by-side comparison, you can see that almost all the proteins fall very nicely on diagonal, indicating that the changes that go on between the two knockouts are pretty much mirroring each other. So the phenotypes are overlapping. However, when we looked at another cell type, these are now embryonic fibroblasts, we saw a very different picture. It turns out this is the most extreme example that we found, but you can see that majority of proteins do not fall on this diagonal. In fact, they fall off this way. A lot of proteins are in fact seem to be, more proteins appear to be upregulated in this Drosha knockout compared to any other situation. So now the reason why we decided to profile these MEFs in the first place was because of another phenotype that we had noticed earlier on. So we'd been interested in the role microRNAs might play in the control of embryonic stem cell differentiation and in reprogramming. And because of a phenotype that we noticed, that we went back and did this proteomic profiling. Anyway, so these experiments, I won't go through in detail, but this basically we're using the protocol, the Yamanaka protocol, which a lot of people now use, the introduction of some reprogramming factors and to track the ability of these factors to convert a somatic cell back into a embryonic stem cell-like state, although not exactly like embryonic stem cells. So just really quickly, so in these experiments, basically we start off with fibroblasts, we introduce these reprogramming factors, and you just track them over a couple of weeks and watch them turn back into something like an embryonic stem cell. And we can then check whether they're really reprogrammed by reactivation of stem cell genes here, and ultimately whether they contribute to a chimera. So this chimera received cells that had properly reprogrammed, whereas this chimera received cells that didn't get reprogrammed, so the cells didn't incorporate, that's why it's white. So anyway, we did these experiments with Drosha and Dicer knockout cells. So first off, when you do these experiments with control cells, you find that about 25% of them are reprogrammed, and the 75% of them simply immortalize. So they immortalize and they grow properly, but they never turn on the endogenous stem cell genes. When we do this experiment with the Drosha knockout cells, we find that you only get immortalization and you never get reprogramming. And then when you do the experiment with the Drosha knockout cells, you don't even get immortalization. In fact, the moment you delete Drosha, the cells senesce and they stop growing and they just sit there on the plate and do nothing. So what we have here is a situation where the deletion of Drosha results in a more severe phenotype than the deletion of Dicer. Here's another example. These are some experiments done by Tim Johansson, a PhD student with Andrew Liu and myself, and he's looking at the effect of deleting these enzymes in dendritic cell differentiation in vitro. So what he's done here is to sort LSK cells from the bone marrow and induce them to differentiate towards the dendritic cell lineage using FLT3 ligand. So after a certain number of days in culture, what he usually sees are these populations here. These are what we would call conventional dendritic cells. These ones here are the plasmacytoid dendritic cells, and these cells here are probably undifferentiated cells. So when he does these experiments with the Drosha knockout cells, he finds that there is a substantial loss of conventional dendritic cells, plasmacytoid dendritic cells, and then an increase in the undifferentiated cells. When he does the same experiment with the Dicer, you can see that there's no effect. So once again, we have another situation where there is a much more severe phenotype with the Drosha knockout than the Dicer knockout. So we've done this kind of Drosha-Dicer comparison across a lot of tissues now, of course with a lot of collaborators, not by ourselves. And in most cases we find, this is a few examples, in most cases the Drosha versus Dicer knockout basically is the same. The two different knockouts get pretty much identical phenotypes. In a number of mice, a number of tissues, we now find that the knockout of Drosha results in a more severe phenotype. And also, in a number of tissues, we find that the knockout DICER results in a more severe phenotype. And I'll come to one of these examples in the last part of my talk. So let me discuss more about this situation where DROSHA deficiency is causing more severe phenotype. So the fact that DROSHA is causing more of a severe phenotype really suggests to us that it must be doing more than just generating microRNAs. Otherwise, the two knockouts would have been the same. So what we thought about was what DROSHA might be doing. So the role of DROSHA is to recognize this stem loop structure within the precursor to ensure that that precursor enters the microRNA pathway. So we asked, what would happen if that kind of structure was present in some other RNA, say in a protein coding messenger RNA? Might DROSHA recognize the structure and then cleave it? So we looked more carefully at the genes that were affected by DROSHA deficiency but not DICER deficiency, and looked at the messenger RNAs with a little bit more detail. And sure enough, we find that at least some of them appear to contain these, something like stem loop structures in the messenger RNA. And you compare this to bona fide microRNAs here, you can see that there are some similarities, so perhaps. So what we needed to do first was to look for evidence that messenger RNA cleavage is happening in a cell. First off, what we did was to mine some old sequencing data we had generated. So these are just deep sequencing data sets of processed RNAs, small RNAs. So these are just RNAs that have been processed by endonucleases and not degradation products in the cell. So we just mine these data sets. So they mostly contain microRNAs, but it turned out that other RNA fragments were also present. For example, these are two genes, DGCR8, SCD2. Here's the putative stem loop stretched out at the bottom, and we see that we find these different fragments coming out in these cells. In this case, we were looking at T-cell progenitors. We also looked at the cleavage of the messenger RNA directly, and again, these are experiments done by Tim Johansson. And what he's looking for is the presence of a cleavage point within a messenger RNA. So cleavage by an endonuclease, such as DROSHA or DICER or any of the others, will leave specifically a 5' phosphate. So by taking advantage of this 5' phosphate in a specific chemical reaction, we're able to identify transcripts that have been cleaved using an adaptive ligation experiment. So he did this in lineage-depleted bone marrow progenitors and found that a number of messenger RNAs do indeed have these cleavage sites. Again, this is an example. This is DGCR8 again. This is another SCR2, and the X's indicate the points in which a cleavage site was identified. So in this case, this is the 5' end of this gene. You see the cleavage sites here in the 5' UTR and 2 in the open reading frame. And in this transcript, we see that the cleavage is occurring towards the end of the gene. Here's the poly A here, and these cleavage sites are within the open reading frame as well. So what we're seeing is that messenger RNAs can be cleaved in cells, but of course this doesn't mean it's caused by DROSHA. We still need evidence that DROSHA is cleaving those messenger RNAs. And we did this in a couple of ways. First off, this is an experiment where we've simply mixed messenger RNA transcripts that have been radio-labeled, so they're radioactive at the top here, and then we mix them with immunoprecipitated DROSHA and then run the cleavage products under the gel to see if we can see any cleavage going on. And you can see here, you can probably just make out a faint band generated here, 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 here. So this appears to be some cleavage of messenger RNAs. However, you'll notice that it's never as strong as the cleavage of a microRNA, but at least we see some cleavage. And then the next step was to confirm that loss of DROSHA will abrogate these cleavage reactions. And these are experiments now done with Greg Hannon's lab at Cold Spring Harbor. And here we've done the same experiment where we've cloned out using adapters transcripts that have a 5' phosphate indicating cleavage. But instead of simply doing a PCR, what we did was to generate next-gen sequencing libraries and then did a genome-wide sequencing. In these experiments, we used embryonic stem cells in which we could induce deletion with tamoxifen, so they carry a CRE in them. So first off, when you do the sequencing experiment, we see that there are again cleavage peaks in the wild-type ES cells, so that's in the blue in these two transcripts. We found, I think it was 8 transcripts that had these peaks. And then when we delete DROSHA, we see that we lose that cleavage product. So confirming that this cleavage is indeed mediated by DROSHA. So up until this point, this has really just been a phenomenon to us. We've noticed it in different cell types. We're pretty confident it was real. In fact, a couple of other groups also detected the cleavage of DGCR8 by Drosha in a couple of other cell types. I think it was in 293 cells, which is a human cell line, and also in Drosophila cell lines. So we're pretty confident this cleavage mechanism was a real phenomenon. Problem was, we had no idea whether it had anything to do with biology. We had never any evidence that it was doing something, other than the fact that there was sometimes differences between the Drosha and DICER knockout. So this was until really the last couple of years, when one of our collaborators stumbled upon a phenotype that appeared to be related to what we were looking for. And I'll show you a few of these experiments. So this is now work done in Vernon Taylor's lab, who was originally at the Max Planck, but has recently moved to Bristol. And a lot of the experiments I'm going to show you in these last couple of slides was done by his PhD student, Phil Knuckles. So what he's looking here is the effect of deleting Drosha or DICER in neuronal progenitor cells. And what he's done is to electroporate in a Cre-expressing plasmid into the forebrain of E13.5 embryos and look at what happens when you induce deletion. And the Cre is under the control of the Hes5 promoter, which drives expression in the neuronal progenitors. And what he found was that there was an effect on the Drosha knockout cells, but not in the DICER knockout cells, and that's shown in this slide. So first off, this green reports for PAC6, which again is another reporter of the neuroprogenitors, and the red, it reports on the transfection of the Cre, so whether the Cre has been taken up. So when he transfects in the Cre, he finds that there are plenty of progenitor cells, neuroprogenitors, that are present in this ventricular zone of the forebrain. So these are the controls. However, when he does the same in the Drosha conditional knockouts, the presence of the Cre now deletes Drosha, and he finds that there are almost no transfected cells in the forebrain. So the cells that have lost Drosha have disappeared in some way, and it almost appears that, I don't know how well this comes across, but these cells are now migrating into this subventricular zone. When he does the experiment with the DICER knockouts, you can see that there are plenty of progenitor cells that have taken up the Cre. So deletion of DICER here has no effect, whereas the deletion of Drosha has an effect. Now, the loss of Drosha has quite a severe consequence. So this is has severe consequence on the morphology of the forebrain. So this is what the cells look like. It's well organized in the wild type situation. These are stained for a few different markers. And when you knock out Drosha, you see the cells become completely disorganized, and basically everything becomes messed up in terms of structure. So there's a severe consequence with the loss of Drosha. So what it appeared to be going on was that the neuronal progenitors were losing stem cell state. What they did was to take out, to generate neurospheres from these mice, the Drosha and DICER knockout mice, and analyze the expression of a number of transcripts. And it turned out there was this massive upregulation in the transcript neurogenin 2. Neurogenin 2 is a marker of differentiation, and these should not be in these progenitors, in these stem cells. And you'll notice here on the left here, there's also a slight upregulation of DGCR8, that target I was telling you about. So given that there was this effect in the Drosha knockouts, but not DICER knockouts, we were wondering whether this may have something to do with our messenger RNA cleavage mechanism that we stumbled across. So we looked a little bit more carefully at the messenger RNA of neurogenin 2, and it appeared that there were what looked like up to 4 potential stem loop structures within the 3' untranslated region of the transcript. So potentially Drosha might be recognizing these transcripts and cleaving them, and it may be this that is regulating its expression or repressing its expression in neuronal progenitors. So to check that Drosha is indeed binding to this, we did a clip experiment. This is an immunoprecipitation after we've cross-linked RNA to proteins. So what we've done is to immunoprecipitate Drosha out of these progenitor cells, neurospheres, and then prepare RNA, and then amplify the RNA to see what's present. And sure enough, we can find that neurogenin 2 is pulled down when we immunoprecipitate Drosha. And as a control, we can immunoprecipitate DGCR8 as well. And then here on the right, this just shows that the messenger RNA, the neurogenin 2, is cleaved, and it's dependent on Drosha. So in control cells, in wild-type cells, you never detect the full-length 3' UTR, so this part here. However, when you knock out Drosha, the 3' UTR now becomes clearly prominent. So what we had here, what we were thinking is that Drosha normally functions in these progenitors to repress neurogenin 2 by cleaving these stem loop structures, and this was how Drosha may be functioning to maintain the fidelity of these neuroprogenitors. So really the final experiment we needed to do was to confirm that if we now de-repress, if it's really the de-repression of neurogenin 2 that was causing this phenotype. So in this last experiment, what Phil did was to force the expression of neurogenin 2. So if the de-repression of neurogenin 2 was really what was causing this loss of neuroprogenitors, if we force the expression of neurogenin 2, we should in theory phenocopy what we saw when we knocked out Drosha. And sure enough, when you transfect in neurogenin 2, we lose the neuroprogenitors, whereas if you do it with a control messenger RNA, you don't lose it. So what we have here was the first time an actual biological function for this messenger RNA cleavage mechanism we found. And we're very excited about this. This was our first evidence, but I suspect we will find others in the future. I don't think this is a restricted example. All right, so this is the second part of the talk. That's all I'm going to show you for this. Basically, what I've told you is that messenger RNAs contain, at least some of them contain secondary stem loop structures that can be directly recognized and cleaved by Drosha. And in most cases, we find that the cleavage of these messenger RNAs by Drosha was actually less efficient than the processing of bona fide microRNAs. So to us, this kind of suggests to us that rather than simply degrading everything, this may be a mechanism by which Drosha fine-tunes expression levels depending on the situation. Of course, the one example that did not fit this was the neurogenin 2, and it's probably why we ended up picking this up so easily, was because this neurogenin 2 was pretty much completely cleaved and degraded in neuroprogenitors by Drosha. All right, so on to the last part of my talk. So the last 10 minutes, I'll tell you a story about DICER. Again, this is a story that involves a function that is independent on microRNAs. Now, this story was actually initiated by J. M. Batty at the University of Kentucky. And actually, a lot of the experiments I'm going to show you again is now done by Hiroki, his postdoc. Jay is actually an ophthalmologist by trade. And Jay's lab has been interested in trying to understand mechanisms that may be involved in various macular degenerative diseases, so diseases that affect the retina of the eye. And it's because of an initial finding that they made that they came to us, and we followed this trail along DICER, and I'll show you where this came from. So one of the diseases he was looking at is known as geographic atrophy. This is a very severe form of macular degeneration. You see this damage to the retina here. It's an untreatable disease. It is age-related. And when they were studying samples from donors, they noticed that one of the genes that was dysregulated was DICER, but not other components on the microRNA biogenesis pathway. And that's shown here. So this is the RNA level. We can see that there was downregulation of DICER here in many of the donor eyes, but not of other components. And down here is the protein level of DICER. You can see that there's a slight downregulation of DICER compared to control, and each of these is a separate donor eye. But of course, these are all done in humans, and there's so much you can do with human donor eyes, which are pretty rare to come by. And given this apparent connection with DICER, this was when Jay contacted us to explore this and to see whether this DICER connection might actually have anything to do with the mechanism of this macular degenerative disease. So the first thing we did was we sent them Drosha and DICER conditional knockout mice, and we wanted to test whether DICER deletion was in fact responsible for any of this phenotype. So we crossed it to BESCRE, which expresses in the retinal epithelium, and this is what they found. So first off, in healthy controls, you can see that at a macroscopic level, the eye is smooth. You can see the structures are all in place. You don't see any major damage to the tissue. And when you zoom in at a microscopic level, you can see that the cells are nicely ordered and organized in the epithelium. These sections have been stained for ZO1, which stains for tight junctions. Now, when you knock out DICER, you can see now that there is the surface of the retina becomes a lot more mottled, and when you zoom in at the microscopic level, the damage is it's very obvious. When he did the same with the Drosha knockout, you see that the eyes were perfectly normal. So here we have a situation where loss of DICER has a strong phenotype, whereas loss of DROSHA does not. So to us, this indicated that whatever DICER is doing, it must, again, not be related to microRNAs. Otherwise, the two would have had similar phenotype. So by this point, we had known that DICER is probably involved in the processing of lots of other double-stranded RNAs, at least in theory anyway. We certainly know that it can process double-stranded RNAs in vitro when we perform experimental RNAi. So one of the things that Hiroki did was to stain the eye sections with an antibody that detects double-stranded RNA. And you see that there's a huge accumulation of the double-stranded RNA in these epithelial cells. He then used the antibody to pull down lysates from epithelial cells grown up in culture, and then he used adapter-ligated PCR, and this amplified what was being pulled down. So these are the different clones that were pulled down, and when we sequenced them, they all turned out to correspond to ALU RNAs. So ALU RNAs are derived from ALU elements. It is a retrotransposon, so it's element that has integrated into the genome. In humans, actually ALU elements are present throughout primates. And shown here is the 46 chromosomes of humans, and these have been hybridized to a probe that detects ALU elements in green. And you can see that the ALU elements distributed throughout all the chromosomes. There are literally thousands of these elements spread throughout the genome of primates. So these elements appear to be expressing RNA, at least in that situation. So we wanted to determine whether it was the loss of DICER that was responsible for this abnormal expression of ALU RNA. So what we did here was to take epithelial cells, human epithelial cells, from the eye, and in these experiments we knocked down DICER using an antisense RNA. And you can see that over time you get this accumulation of ALU RNA in the cells. On the right here is an experiment done with now mouse retinal epithelial cells. What we did was to take epithelial cells from DICER conditional mice and then deleted DICER with an adenovirus Cree. Now mice do not, the mouse genome does not have any ALU elements, they're specific to primates. Instead, rodent genomes are infestated with another group of retrotransposons, which are quite similar to ALU elements. These are B1 and B2 elements. And when you do this deletion in the mouse, again we see that these retrotransposon-derived RNAs are upregulated, so that they're de-repressed. So what we're thinking now is that perhaps DICER was recognizing these RNAs and actually directly cleaving these RNAs in cells, at least in healthy cells. And that's finally shown here. On the right here is a prediction of the secondary structure of a human ALU RNA. And when you fold the RNA in silico, you can see that there is a lot of predicted secondary structure going on here. So we suspected that perhaps DICER could recognize these stretches of double-stranded RNA and then cleave the RNA. And sure enough, when we mix in vitro transcribed RNA with DICER, we see that they're cleaved. So in this first lane is just the ALU RNA alone. The second lane is when we mixed DICER in, and you see that the RNA is now cleaved. And then this final lane is when we denatured DICER. So the DICER is heat sensitive. So our hypothesis then became that in diseases such as geographic atrophy, the loss of DICER may allow the upregulation of what might be cytotoxic RNAs, and it's perhaps these RNAs that are what's causing the damage. So our final experiment that we needed to prove this was to now see if we can now reintroduce ALU RNAs back into the eye, or in the case of the mouse, whether we can introduce B1 and B2 RNAs back into the eye and see whether we can recapitulate this macular degenerative disease. And that's shown here in this last experiment. So here, Hiroki did an experiment where he's now injected a plasmid encoding B1 and B2, so the equivalent, mouse equivalents of the ALU RNA, and injected it into the subepithelial layer of normal wild-type mice. And when he does that and ectopically enforce the expression of B1 and B2, you see that the epithelial layer again gets damaged. So we have a situation here that it's the ALU RNAs that are directly cytotoxic to the retinal epithelium. All right, so that's all I'm going to show you for this last part. So what I've shown you is that DICER, at least in the eye, is normally involved in degrading ALU and other sign-derived RNAs. And the loss of DICER in diseases such as geographic atrophy results in accumulation of these otherwise cytotoxic RNAs, and it's these cytotoxic RNAs that go on to destroy the epithelial cells within the retinal epithelium. Jay's group has now gone on to work out what the mechanism of this damage is, and it appears that the accumulation of RNAs is actually activating the immune response in the eye, and that is what ultimately destroys the eye. But that's really another story. So finally, what I've shown you is a situation that we found in which DICER deficiency, but not DROSHA deficiency, causes a phenotype. All right, so that's pretty much all the data I'm going to show you today. So I hope I've left you with the impression of just how complex and interesting the biology of RNase 3 enzymes is. You know, really until recently, when we talked about DROSHA and DICER, we pretty much only thought about it in this pathway, in the microRNA pathway. We really only thought of it as being central to microRNAs. Not to say it isn't. I mean, they certainly are central to the microRNA pathway. And I showed you one, and I talked about one of my stories in which DROSHA is involved. However, I've also shown you that DROSHA is involved in recognizing and cleaving messenger RNAs that contain the secondary stem loop structure. And it appears that this is a mechanism of fine-tuning or repressing messenger RNA expression that should not be occurring in a cell. I've also shown you that DICER has another function in degrading cytotoxic RNAs. And of course, DICER also has a function in producing siRNAs when we perform experimental RNAi in the lab. Now, I didn't talk about any of these other things, but it turns out that this whole system is even more complex. We've also identified different classes of microRNAs that bypass DROSHA and function in other pathways. We've also discovered a microRNA that bypasses DICER and goes straight on from DROSHA. So this whole pathways are certainly a lot more complex than we ever imagined. And of course, I haven't even talked about the potential role of MRPL44, the third RNase 3 enzyme that perhaps fits somewhere in this row. All right, that's pretty much where I want to leave it. Before I stop, I'd like to thank people who've helped out in any way. This is the group at the moment. This is the current group. I really didn't talk too much about their work, other than showing a few experiments from Tim Johansson, a PhD student with Andrew Liu and myself. For the first story, most of that work was actually done by two former RAs. Francis did most of the computational biology, and Ken did most of the bench work, with some assistance from Lachlan, who was a Europe student the first year I was back, and also Fiona did a few experiments. The work I showed you, I mean, you can imagine, I told you a whole diversity of different projects, and really they've only been possible because of some really fantastic collaborations. So the second story, the work on the cleavage of messenger RNAs, was actually initiated while I was still a postdoc with Dan Lippman at New York University. And the SILAC experiments, the proteomic profiling experiments, were actually done with the help of Tom Newbert and his postdoc, Guan Zhang, who are also at New York University. For the second story, for the first story even, the mapping of microRNA gene structures, we've had a lot of help from Rick Myers and his group at Hudson Alpha. They were instrumental in helping us get all our RNA-seq libraries going when we first moved back here because, well, the AGRF wasn't up and running yet in terms of sequencing, and we still hadn't sent our libraries off to BGI, but we're of course doing that now. The messenger RNA cleavage story has really been a fantastic collaboration with two groups, Greg Hannon and his lab at Cold Spring Harbor, and Verdun Taylor at the Max Planck, who was at the Max Planck Institute, but has just moved to Bristol University. And that last story was on the macular degeneration, was initiated by J.M. Batty at the University in Kentucky, and we've had the privilege of being intimately involved with the molecular work on that aspect. And finally, I'd like a huge thank to Len Harrison and Andrew Liu and their respective lab members. They were extremely helpful with helping me get set up again 3 years ago when I first moved back to WEHI. And of course, where would we be without all the funding that we've received from all these important organizations? Thank you. I'll stop there and take questions.